Hi there, and welcome to this video on GCSE Biology for the AQA Specification, focusing on photosynthesis, and in particular on the rate of photosynthesis. I'm Shumina from StudyMind, where we help you revise GCSE Biology with our helpful video tutorials, tailored to your subject, your specification, and to you. If you're new here, make sure you click the subscribe button. Whilst you are watching, please leave any comments below if you're unsure about anything, and let us know if it's your first time watching our videos so we can send you our free revision materials. We also have helpful timestamps below for each part of the video to help guide you through the specification. So let's get started. Hello, and welcome to this tutorial on photosynthesis. And in our second lesson of 3 sessions on photosynthesis, we're going to be having a look at the rate of photosynthesis. So in our previous session, we had a look at the basic photosynthetic reaction. And so in order to do that, we had a look at the word and the chemical equation. So do go back to that because it's really important that you understand exactly what makes up the components required for photosynthesis in order to understand this tutorial. So today we're going to be having a look at the factors affecting photosynthesis. We're going to look at how we can measure the rate of photosynthesis using the Canadian Pondweed experiment, and we're going to have a look at how we can use the inverse square law in this. And finally, we're going to have a look at how farmers might control rates of photosynthesis in order to optimise their yields of their crops. And so we're going to have a look at greenhouse conditions. So first of all, let's have a look at the varying effects of limiting factors on the rate of photosynthesis. So I just use the term limiting factor. So a limiting factor is a condition that, when in shortage, slows down the rate of a reaction. So it limits the reaction, hence it's a limiting factor. And so light intensity, carbon dioxide, whoopsie, went a bit too fast there. Light intensity, carbon dioxide concentration, and temperature are all limiting factors for the rate of photosynthesis. So that means that each of these factors, individually and in concert together, can affect the rate at which a plant undergoes photosynthesis. So let's break this down a little. So let's have a look at what this graph is showing us. So we have carbon dioxide concentration on the x-axis, so the more we move this way, the higher the concentration of carbon dioxide, and we have the rate of reaction on the y-axis. So this is how you should always start to analyse a graph. Have a look at what exactly it's trying to tell you. So here we can say that it's measuring how carbon dioxide concentration changes and how the rate of reaction changes. And since this is on the x-axis, your carbon dioxide concentration is going to be your independent variable, whereas the rate of reaction is on the y-axis, so it will be your dependent variable. So the rate of reaction is going to depend on your carbon dioxide concentration. So that's really how I'd best advise you to analyse graphs, is have a look at your x-axis, your y-axis, determine which is your dependent and which is your independent variable. And now we can have a look at what it's actually showing us. So this is saying we're increasing the carbon dioxide concentration and we're also increasing the rate of reaction at the same time. But this effect is going to plateau. So there's going to be a point where we increase the carbon dioxide concentration. So I would say from around this point onwards, we are increasing the carbon dioxide concentration here, but we're getting no change in the rate of reaction. We can say it's staying more or less constant. So we would call that that disappeared. We would call that a plateau. So here, where your carbon dioxide concentration is increasing, but your rate of reaction is not increasing, we would call that a plateau. So just to break that down a little more, let's have a look at this in further detail. So as I said, the higher the CO2 concentration, the higher the rate of reaction until we get to a certain point. And then at high CO2 concentrations, the rate levels off, it plateaus. And why does this happen? it's because increasing the carbon dioxide concentration further is not going to have any effect on the rate of reaction because the enzymes required for photosynthesis are saturated. This means they're being fully worked. They can't physically work anymore. Even if you increase the carbon dioxide concentration, it's not going to benefit the rate of photosynthesis. So just to summarise what I said, here are the main points. Oopsie. So what you need to gather from this is that there's a section of the curve at which carbon dioxide concentration is the limiting factor. And how do I know this? Well, because if I'm increasing the carbon dioxide concentration at these points, the rate of reaction is also increasing. We're getting this increase in the rate of reaction as carbon dioxide concentration is increased. So that occurs up to about this point. And here, I'd say, so from this point onwards, I'd say CO2 is no longer the limiting factor, so something else is limiting the rate of reaction from that point onwards. So now let's have a look at this in the same way, but with light intensity. So again, look at this graph. Try and sort out what's on the x-axis, what's on the y-axis, and therefore what's your dependent variable and what's your independent variable. So I'll just give you a couple of seconds to do that. Okie dokie, so your light intensity is on your x-axis, so that's going to be your independent variable, you're controlling it. Your rate of reaction is on your y-axis, so that's your dependent variable. You're seeing how the dependent variable varies as you increase or decrease the light intensity. So that's that sorted out. We can now move on to what the graph is telling us. Well, as we get an increase in light intensity, we're getting an increase in the rate of reaction, up to a certain point again, so we can see the curve begins to plateau. So analysing this in the same way as we did with carbon dioxide, we're saying the higher the light intensity, the higher the rate of reaction. And why does this happen? Well, it's because more light at higher intensities can be trapped by the chloroplasts. And at high light intensity, the rate of reaction levels off, so it plateaus again. And that's because the maximum amount of light has already been trapped. We can't physically trap any more light, and so something else is now limiting. So as with the previous example, we have light intensity limiting at these points, and we have another factor, so sorry, I'll just write light intensity limiting, so that's your limiting factor, whereas here, something other than light intensity is limiting. So I've already said that from this point onwards, the maximum amount of light has already been trapped. So perhaps a way of increasing the rate of reaction further is by providing more chloroplasts, because then more light can be trapped at these higher light intensities. So, for example, chloroplast number could be a limiting factor around here. So again, this just summarises what I just said. Now let's move on to the effect of temperature. So again, do the same as before, decide your dependent and your independent variables, and I'll just give you a few seconds to do that. So your independent variable is the temperature on the x-axis, your dependent is the rate of reaction on the y-axis, and I know that because usually the independent variable goes on your x and your dependent on your y. So this means that we're controlling our temperature and seeing how the rate of photosynthesis varies with this. So we can see that the higher the temperature, the higher the rate until a certain point, because the enzymes are gaining more kinetic energy from the increasing temperature. Now notice here that we don't hit a plateau. We hit an optimum rate at the optimum temperature, so there is a temperature at which our rate of photosynthesis is highest. Now there's no plateau after that. Remember with the other graphs, they kind of went like this, didn't they? With light intensity and with carbon dioxide concentration. But with temperature, we have this steep decline. Now, in order to understand why this happens, you've got to think about enzyme kinetics. So remember that at high temperatures, your enzymes denature. Their protein structure is going to break apart because we're breaking up these weak hydrogen bonds that hold it together. So if your enzymes are denaturing, you're not going to have anything catalysing your reaction, so your rate of reaction is going to fall. So that's the key difference with temperature, and you've really, really got to look out for this, because in the exam they might ask you to reproduce these curves or explain them, and if you just remember that with carbon dioxide and light intensity you get that lovely rise and then plateau, but with temperature, because of your enzymes denaturing, you're going to get this fall. And I think, in my opinion, if you understand why this is happening, it's not so much about memorisation, it's just about understanding the process, and therefore you're going to be able to draw this curve in the exam. So if you don't quite understand what's happening at the moment, go back and watch these last few slides again, because once you gain the basic understanding, it's going to be really, really easy for you. And this again just summarises what I just said. So we've covered carbon dioxide concentration, light intensity and temperature, and now we're going to have a look at another limiting factor, so chlorophyll. So chlorophyll can act as a limiting factor for photosynthesis. As the amount of chlorophyll increases, we're going to be able to trap more light by the chloroplast. However, once there is a certain amount of chlorophyll, it will no longer be a limiting factor. So now let's have a look at some graphs that include all of this information on one diagrammatic format. So here, again, have a look at what's your independent, your dependent. Well, you'll be very well practiced with this now, so I'll go straight on to it. So light intensity is your independent variable, your rate of reaction is your dependent. What else are we measuring on this graph? Well, we can see that we're varying the temperature. So if this confuses you slightly, just block out the 30 degrees Celsius line for now and have a look at the 20 degrees Celsius line. So what we would be investigating with that is how light intensity affects the rate of reaction at this set temperature, and again at this set temperature. So the difference between these two curves is basically showing the difference between these two temperatures. So we can see that as you increase light intensity, the rate of reaction increases until we reach a plateau, and this occurs at both 20 degrees Celsius and 30 degrees Celsius. And as you increase the temperature, the rate increases. So when you increase it by 10 degrees, the rate at any respective point on these curves, so if we take this point of light intensity, we're going to get an increase, sorry that was a bit wonky, we're going to get an increase in the rate of reaction. That's our increase in the rate of reaction there as we increase from 20 degrees Celsius to 30 degrees Celsius. And we can notice here that the light intensity affects the plateau at a higher level. So the light intensity is a limiting factor for longer, as opposed to temperature. So again, this just summarises what I just said. And now notice in this graph we kept CO2 constant. Now we're going to keep temperature constant but vary CO2 concentration. And again you can see a similar thing. So as light intensity increases, the rate of reaction increases until we reach a plateau. And as we increase carbon dioxide concentration from 0.05 to, whoopsie, I didn't mean to draw on that, from 0.05% to 0.5%, we're going to get an increase in the rate of reaction too. And we can see that the light intensity affects the plateau at a higher level, so it is a limiting factor for longer, again. So this, again, summarises what I was just saying. Now, these last few slides that we went through were very dense. Please do go back and watch them because these are going to come up in your exam, and if you understand it, it's going to be really easy marks, but I think it just takes a little bit of time to understand it. So do go back and listen again if you're not quite clear about it in your head right now. So next we're going to have a look at how we can measure and calculate rates of photosynthesis. So let's have a look at this Canadian pondweed experiment. So we have a light source. The light source is going to be positioned a particular distance, which we'll measure with a ruler, from a beaker. And the beaker is going to contain our Canadian pondweed. We can see bubbles of gas that is produced by this pondweed as it photosynthesizes. And if you think back to the photosynthesis equation, that's going to be oxygen bubbles of gas, because that's a product of photosynthesis. And we're going to measure this oxygen production here in this capillary tube. And in order to do that, we're going to measure the movement of this droplet of water along the capillary tube. And basically the distance that the droplet of water moves is going to be proportional to the rate of photosynthesis. So the more photosynthesis that occurs, the more oxygen produced, and that oxygen gas is going to push this along the ruler. And we can vary the light intensity by moving the lamp nearer or further away from the test tube. And notice how we can measure this distance with this ruler here. So that's our Canadian pondweed experiment. So this just summarises it for you. And just remember that the rate of oxygen production is proportional to the rate of photosynthesis. Now, why is that? Well, again, as I said earlier, think back to your equation for photosynthesis. The rate of oxygen production is going to be proportional to the rate of photosynthesis because oxygen is a product of photosynthesis. So if we want to investigate the effect of certain variables on the rate of photosynthesis, we can use this experiment by, for example, affecting the light intensity. To do that, we can place the lamp at differing distances from the plant and measure this distance. So, this would be distance 1, for example. This would be distance 2. And, sorry, kind of going off the page, but that would be distance 3. And then you would compare all three. And it's really, really important to make sure that you control the other variables, because if you're not controlling the other variables, then you're not only investigating the effect of light intensity. So, for example, if you were varying the temperature as well as light intensity, whatever kind of rate of photosynthesis you're measuring will be a product of the change of that light intensity and the change of the temperature. So you've got to keep the temperature constant while you vary the light intensity. It's also really important that you repeat the experiment 3 times at each distance, and that's in order to increase reliability of your results. So in a similar way, you can investigate the effect of temperature. But in order to do this, you would perhaps place the boiling tube in different temperature water baths, so a 10 degrees Celsius water bath, a 20 degrees Celsius water bath, and a 30 degrees Celsius water bath. And you would then observe the oxygen production through the movement of the bubble along the capillary tube, and that would be proportional to your rate of photosynthesis. And if you compare these 3 results, you can then compare the 3 rates of photosynthesis. But again, you're going to have to control the other variables and repeat this 3 times, taking a mean of the 3 results you get. And remember, your mean is just your average. And again, for carbon dioxide concentration, so this is a little bit more technical because it's a little bit harder to control your carbon dioxide concentration in the environment. So to do this, what we do is we dissolve different amounts of sodium hydrogen carbonate in different boiling tubes to see the effect of carbon dioxide concentration. So the more sodium hydrogen carbonate you put into a test tube, the higher the CO2 concentration, the lower the CO2 concentration. And again, you're going to control your other variables and repeat 3 times at each distance for reliability, taking a mean when you're plotting up your graph. So can you see a really, really common trend going on here when we're investigating the effect of limiting factors on the rate of photosynthesis? Well, what we're doing is we are varying the independent variable, so that would be your carbon dioxide concentration in this case, and we're controlling all the other variables in order to keep it a fair test. And at the same time, we're repeating in order to make our results reliable and reproducible, and we're taking a mean of these repeat values. So those are your trends there. And so if you ever have to describe an experiment, to measure the rate of photosynthesis in an exam, you've got to remember that you have to do certain things to vary your independent variable, your CO2 concentration here. You've got to control your other variables for a fair test, and repeat, finding a mean when drawing graphs. So those are your key things to remember. And this does come up quite a lot, so make sure you commit this experiment to memory, the Canadian Pondweed experiment. And here are some key rules of plotting graphs, which you might have to do in the exam. So label the axes, put in the units, and take up at least half of the space on the axes with the graph. So this means that here you would have to label this graph. So on the x-axis you're going to have your distance, and I know that's on the x-axis because that's our independent variable, and I imagine that would be distance of the light source from your plant. So that's going to be proportional to your light intensity. And on your y-axis you're going to have number of bubbles. So number of bubbles. And because that's a number, it has arbitrary units, so you don't have to put them in. And you might even want to, on your axis, when you draw this line, you might want to put in your x and your y. So, now let's have a look at the inverse square law. So, earlier on I was talking about how the rate of oxygen production is proportional to your rate of photosynthesis. So that means as your oxygen production goes up, your rate of photosynthesis goes up, or vice versa. Now we're talking about inverse proportion, so it's going to be the opposite of that. So when one variable goes up, the other variable goes down. So for example, light intensity is inversely proportional to the square of the distance. So that means as you increase your distance, you're going to get a decrease in light intensity. And the way we write an inverse proportion is like this. Oops. Sorry, going a bit too fast. It's like this. So light intensity is proportional to 1 over the distance squared. And you can just use this like any old equation. So don't get put off by this little proportion sign here. So you would say, if I write it like this, light intensity is proportional to 1 over distance squared. So for example, what is the light intensity if the distance is 2 centimetres? So you would just plop it into this equation. So the distance is 2 centimetres, so you'd replace that with d. So 1 over 2 squared, which is equal to 1 over 4, which is equal to 0.25. So that's your light intensity. So that's really, really simple, which means you just have to commit a couple of things to memory. This equation here, and understand that you would just substitute in the distance into that equation. Now, last of all, let's have a look at how farmers might optimise their farming conditions for their plants. Because remember, at the end of the day, we want to optimise our yields. So greenhouses can control various conditions. So temperature can be controlled via the glass exterior, which helps to trap the sun's heat. Similarly, blinds can be used if it's too hot. And heaters can be used to provide artificial heat in the winter months. Because remember, we want to regulate our temperatures that it's at its optimum in order for photosynthesis to take place. And we also want to control the other limiting factors for photosynthesis. So think back to those graphs we had a look at earlier on. So apart from temperature, we also looked at the effect of carbon dioxide concentration. And greenhouses can indeed burn paraffin heaters to give off carbon dioxide and therefore control carbon dioxide concentrations within that environment. Also, we had a look at how light intensity can affect the rate of photosynthesis. So light can be artificially provided to provide the energy required for the photosynthesis reaction. And overall, we're going to have to really carefully monitor these conditions because you've seen those graphs. You've seen how a small change in your limiting factor can affect your rate of photosynthesis. So you're going to want to really carefully monitor your light intensity, your carbon dioxide concentrations, and your temperature in order to provide the greatest amount of produce for the money put in, so in order to maximise your yields. And also think back to the other graph we had a look at. Oh no, it wasn't a graph, we just mentioned that another limiting factor was your chloroplasts and your chlorophyll concentration. But that's not really something that farmers can very easily regulate. But remember in the last tutorial we saw how some diseases might actually cause lack of chlorophyll production, and therefore this will also affect your rate of photosynthesis. So overall, farmers can control their carbon dioxide concentration, their light intensity and their temperature in their greenhouses, and this is utilised in order to maximise their crop yields. So well done for today. We really did cover a lot of dense stuff, so go back and look at those graphs and really crack them and understand them, perhaps even draw them out, explain them to yourself the same way that I explained it to you, and I really hope that helped. So well done, and I'll see you for the next session. Thanks for watching this free video from StudyMind. If you liked this video, make sure to subscribe to catch our newest videos by clicking below, and leave a comment on a topic you'd like a video on. Click here to watch more videos in our series for GCSE Biology, or visit our website studymind.co.uk for free past paper compilations by topic and specification.